Hi, everyone. We're ready to take a look at Chapter 1 in our textbook, whose title is Introduction to Human Anatomy and Physiology. If you open your book to page 9, you'll see the beginning of this chapter, which covers a variety of topics, sort of general topics that pertain to science. What is ANP? A little bit of history behind the human body in terms of how discoveries were made, primarily due to dissections that were done in the Middle Ages. So I'm going to kind of cover a couple different topics that this chapter talks a little bit about. We won't talk about every single section, but I do want you to read it. It's a very nice introduction, as the title sort of implies. What I'd like to do is to share my screen and take a look at the PowerPoint that accompanies this chapter. Remember, when you go into Blackboard, in the contents section of the course shell, you should see chapter PowerPoints listed. And I'm going to be using these for all of the chapters in lecture. So you're going to want to make sure you're looking at those. Some people like to print them off in such a way that they can write notes next to the actual slide. So you can certainly do whatever you'd like in terms of how you want to take notes. But I'm going to go ahead and share my screen. I'm going to go to the PowerPoint, which is right here. It's a rather short PowerPoint. This first chapter, I think, is only like 18 or 20 slides. And it, of course, begins with this question: what is anatomy? Well, I like to think of it as structure, right? The structures of the body. Those could be bones, those could be muscles, those could be organs, those could be blood vessels, you know, things that we can actually point to physically and say, this is the humerus bone, or this is the biceps brachii muscle, or this is the pyloric sphincter, you know, that leads from the stomach into the small intestine. Of course, another good question to ask is, since we're taking anatomy and physiology, what is physiology? How does it differ from anatomy? How are the two related to one another? Well, if anatomy is structure, think of physiology as function. So they are indeed related. Structure is impacted by function, and function is impacted by structure. They both go hand in hand. A good example I could give you of the relationship between anatomy and physiology is that of your heart. And if you think about the four chambers of the heart, the two atria and the two ventricles, when you look at the thickness, for example, of the left ventricle and compare it to the thickness of the right ventricle, you'll find that it's thicker. The left ventricle has a thicker wall than the right ventricle. And does anybody know why that is the case? Its structure impacts its function. Well, having a thicker wall means that when that left ventricle goes to contract, it pushes that blood out of that chamber with more force, under a higher pressure than it is coming off of the right ventricle. And the reason for that is the blood coming from the right ventricle is going a very short distance to the lungs. Now it's being pumped under pressure, but not as high a pressure because we're not having to go so far. The blood has to travel a relatively short distance to the lungs, as opposed to the blood in the left ventricle that has to be pumped throughout the entire body. So it has to be obviously pumped under a much higher pressure. So there's a good example again of how structure impacts function. And we will be giving you lots and lots of examples of structure-function relationships in ANP 1, and you'll be hearing more about them, of course, in the second semester, ANP 2. We can sort of think of the organization of the human body as being composed of various levels or strata. And so what this slide sort of implies, and we're going to work from left to right here. On the left, we have something called an atom. And you're going to be reviewing atomic structure in chapter 2 as you prepare for that chemistry exam in a couple of weeks. But there are lots and lots of different kinds of elements. You all have heard of the periodic chart, for example. Each of those elements has its own atomic structure. And so what is an atom? Well, it's basically composed of a nucleus, right, that contains protons and neutrons. And spinning around that nucleus are subatomic particles called electrons. And those different numbers of subatomic particles, electrons, protons, and neutrons, vary from atom to atom, from element to element. And that's what gives each element its own unique chemical properties. It has to basically do with the number of these different subatomic particles, protons, neutrons, and electrons. And when we begin to combine different atoms together and form bonds between those elements, those atoms, we form something called a molecule. Now, this particular molecule is actually depicting water. And so chemists like to sometimes color code some of these atoms. And the size of the spheres here also has something to do with the relative size of the atoms as well. And so this is oxygen here in red, and the white spheres represent hydrogen. So two hydrogens and an oxygen, H2O, you know, makes water. So that would mean that this particular atom that we talked about earlier would be an atom of hydrogen, right? Because it's white. So this is a very simplistic molecule made up of simply 3 atoms bonded together. And the particular bond that holds these 2 hydrogens to the oxygen is referred to as a single covalent bond. That may sound familiar from chemistry, high school, or maybe you took allied health chemistry or introduction to chemistry. But chapter 2 will review different types of bonds. This next diagram, the macromolecule, macro means big, right? This is composed, of course, of lots of hydrogens, it looks like, and lots of oxygens. And then we have some other blue colored atoms here, which are, again, different types of elements. This is depicting the macromolecule DNA. And you've all heard of DNA, deoxyribonucleic acid. It's what contains genes that give us particular traits like eye color. Maybe it predisposes us to alcoholism or Parkinson's disease. You know, there's a lot of very interesting traits that we can trace back to the presence of specific genes, they're called, within this DNA molecule. So we'll talk more about that coming up in, I think it's chapter 3. And so I hope you've begun to see that as we have gone from atom to molecule to macromolecule, we're getting bigger, more complex structures, aren't we? So as the red box here is implying, complexity increases as we follow these arrows, eventually making our way to the organism, which is, of course, you and me. But let's go back to the macromolecule, continue working our way up these various strata. A more complex structure than even a macromolecule is something called an organelle. And again, I think you've all studied organelles in high school. This happens to be the mitochondrion, sometimes called the powerhouse organelle. It's here that the cell makes copious amounts of energy in the form of ATP, which stands for adenosine triphosphate. You'll be hearing more about that coming up as well. And the structure of this organelle impacts its function. Just a quick sideline. This structure you're seeing here in sort of yellow is what's called the inner mitochondrial membrane. And you might wonder, why is it folded like this? There's a lot of infoldings. This is to increase the surface area for reactions that are taking place here in the mitochondria that results again in the formation of this energy that cells need. If they run out of energy, the cells die and the organism can die. So this is one of many organelles. You've probably heard of the nucleus, of course, and the smooth and rough endoplasmic reticulum. We also have other organelles like lysosomes, Golgi apparati, microtubules. There's a whole host of different organelles that you're going to be reviewing in chapter 3, actually. As we work our way up from organelle now, we're encountering, of course, a generic cell. And inside that cell, we see not only some mitochondria here, these 6 kind of kidney bean structures are mitochondria. This blue guy you should recognize, of course, as the what? Nucleus, right? And then we have an outer cell membrane, which sort of is like, quote unquote, the skin of the cells, the outermost covering. And we'll again talk more about what that's made up of at the beginning of chapter 3, coming up in the next lecture. From cells, we come to tissues. So, tissues are basically composed of aggregates of cells that have particular functions. And we're going to spend chapter 4 talking about tissues. The whole chapter is devoted to tissues. So we're going to be working our way up this hierarchy, really, in the first few weeks of class. This, of course, refers to the stomach, made up of different kinds of tissues. And, of course, as we look at the next level, we have an organ system. And of course, you recognize it, I hope, as the digestive system. So we have not only the stomach, but we have the liver. Here's the gallbladder in green. This is the colon or large intestine, sort of in a darker orange. And then this pink, kind of centrally located, very coiled structure is the small intestine. We have many organ systems within the organism, right? We have, I think, about 10 or 11 different systems. And we'll talk a little bit more about those and review those toward the last part of this chapter. But you know, of course, reproductive system, muscular system, skeletal system, endocrine system, and so on and so forth. So again, think of the human body as being composed of a number of levels of organization, each getting more complex as we go from the subatomic or atomic level all the way up through the organism level. The book goes into some core themes in A&P. I'm not going to really talk a whole lot about all of those. I think they're quite self-explanatory. So I'm going to just ask you to read over this section here, page 14 through page 16. Also gets into some characteristics of life and requirements of organisms. But what I would like to spend a little bit of time talking about next is this term, homeostasis. This is a term that we will be talking a lot about. We'll be giving you lots and lots of examples of homeostasis in A&P 1 and A&P 2. You can think of homeostasis as the body's ability to maintain a stable internal physiologic state when there are external and internal changes that can sometimes occur. So it's this sort of Zen state. I don't know how else to describe it other than it is a state that your body is trying to maintain as we speak. Because if we were to leave that stable physiologic state, that could be deleterious to our health, could cause real problems. And I'm going to give you some examples of homeostasis next. All of these mechanisms that we'll be talking about involve some players. And here's the first player. We call them receptors. Now, there's a lot of different kinds of receptors in your body. There are touch receptors. There are photoreceptors in your retina. There are taste receptors in your taste buds. You have little hearing receptors, if you will, in your inner ear. We have pressure receptors in our body that can maintain, or I should say monitor, not maintain, but monitor things like blood pressure. When the blood pressure falls or goes up, these receptors become stimulated. When light hits your retina, the photoreceptors become stimulated. So when there are changes that are taking place, now of course it says here too, the internal environment, and typically that's what we often talk about is internal changes, but they could also pick up external changes. For example, when you go outside and it's very, very hot and humid and the sun's shining down, you're going to get hot, right? There are thermoreceptors in your skin too. Okay, so while we often think of internal changes, and most of them are, we could also have external changes too. And think of body temperature changes, for example, as a result of an external change that could stimulate receptors. Okay? So these receptors pick up on stimuli that are occurring as a result of changes taking place in and outside our body. Those receptors are connected to nervous tissue. We'll just say nerves for now. And those nerves will relay nerve impulses from the receptor that's been stimulated due to the change taking place in our body or outside our body. And those impulses are sent to some control center. It is often the brain, doesn't always have to be, but we'll think of the control center in our example for now as the brain. It often is, but there can be other control centers too in the body. And that control center has a particular set point that is going to compare with the change that has occurred. And let's assume that that set point is not the same as what is occurring inside or outside our body. Say body temperature goes up. Now, what's your internal body temperature typically? Anybody know? Your body temperature is about 98.6 Fahrenheit or 37 degrees Celsius, right? So if you go outside on a hot July day, play tennis or run, you begin to perspire. Your body begins to heat up because of the effects of the sun, plus your muscles are contracting and you're perspiring as a result. Well, your body temperature, the thermostat, if you will, in the brain has increased as a result of the exercise or just sitting out in the sun. Normally it's 37, right? So the control center is comparing that rise in body temperature to 37, the set point, if you will, for body temperature. And in an effort to bring that body temperature down to 37, the control center will send impulses via nerves, just like the receptors initially did, right? Only in this case, those nerve impulses will go to effectors, things that can cause something to occur to help us cool down. Now, you see here muscles or glands listed, and those are often important effectors. There could be other examples of effectors too, but we'll stick with muscles and glands for right now. And so in the case of body temperature, when you get warm, as we said a few moments ago, you begin to perspire, you begin to sweat, right? The sweat glands become active. The reason for that is when you begin to sweat, you can more efficiently cool your body through evaporative cooling. And that cooling is what is the response, right? And that will hopefully bring our body temperature down, maybe to 37, maybe not, depends upon how much we sweat and how much we're still running. But the temperature should begin to drop as compared to what occurred prior to beginning to sweat, right? So when the body temperature begins to drop, there's less and less stimulation of those thermoreceptors in the skin. And then there's less stimulation of the control center, which in that case, remember, is the brain, which means that over time, the sweat glands will begin to produce less and less sweat because we're beginning to get closer and closer to that 37 degrees Celsius or 98.6 degree Fahrenheit body temperature. So the takeaway message of this slide is, regardless of the homeostatic mechanism that we want to talk about, all homeostatic responses employ these three players: receptors that detect the internal or external change, the control center, which is stimulated neurally by the receptors, where a set point is compared with the change, and then the control center responding by sending out nerve impulses to some effectors that can affect a change, can cause a response to correct ultimately that deviation in that homeostatic level or set point. Here's a simple analogy that we can use. Think about your thermostat in your house. And let's pretend you have a central air unit outside your home. Of course, you all have a furnace. So in the wintertime, as the temperature drops outside, and we'll assume the thermostat is set at 68, as it implies here on the diagram, as that room temperature begins to fall because it's cold out, the thermostat in the living room or wherever it is going to detect that drop in room temperature. It's sort of like the receptor, the electronic receptor in this case. It sends a signal through a wire to a control center, which in this case is your furnace, and the furnace kicks on. Starts to burn the propane, for example. Heat comes out the vents, or water goes through the hot water baseboard, depending upon your heating unit system. And what happens to the room temperature? It starts to go up, right? Now, when that room temperature gets to 68, are we going to continue to stimulate the furnace, if you will? The answer is no. The furnace will kick off once we get to that homeostatic 68 degree. But if it gets cooler than 68, we continue that loop. On the opposite end of the example would be in the hot summer months, assuming you again have air conditioning. When that room temperature gets above 68, assuming you set it at 68, the thermostat detects that rise, sends a signal through wires to your air conditioner. The air conditioner kicks on, blowing cold air into the room, lowering that temperature until it gets to 68, and then the air conditioner kicks off. So remember, again, the homeostatic level we want to maintain in this case, in our example, is a 68-degree room temperature. If we get too hot, we want to cool it down. If we get too cold, we want to warm it up. Applying that to the human body, we can talk about, again, the role that thermoreceptors play. Those could be in our skin. There are also thermoreceptors in our body as well. And when you find a rise in body temperature, because again, you're running, you're out in the hot sun, as we talked about just a few moments ago, those thermoreceptors detect the change. They send nerve impulses to the control center, which happens to be a part of the brain called the hypothalamus, which compares it to the 37 degrees Celsius body temperature, because that's normal. That's where we want to be. And it, in turn, will stimulate our friends, the effectors, including the sweat glands like we talked about earlier. But also, as you note here, even blood vessels here on the surface of the body, says here they dilate. Why would you expect dilation of surface blood vessels, meaning they get bigger? This is the cross-section of the blood vessel, let's say, on your arm, just under your skin. If it gets bigger, why could that help cool the body? Well, it can help cool the body because most of the heat in our body is carried by our blood. If we can dilate surface blood vessels, we can more efficiently release the heat. That's why the peripheral surface blood vessels will dilate while the core blood vessels actually contract, pushing the blood toward the surface so we can more efficiently get rid of that heat, because that's the goal here, right? Bring the body temperature down. We also, of course, have our sweat glands producing sweat to help with evaporative cooling. And those processes continue until eventually we bring that body temperature down to the desired 37 degrees Celsius, at which case we can then not stimulate those receptors. They don't need to be stimulated because we're at where we want to be. And so this sort of begins to shut down. In the wintertime, when we get too cold, again, you can read through this series of loops, same sort of idea. Receptors pick up on this temperature drop. The hypothalamus compares it to 37 Celsius. And you can read about how our body can generate heat, conserve it in the core of the body, all in an effort, again, to maintain that important body temperature of 37 Celsius. Now, we have been talking a little bit about a mechanism here that employs what's referred to as negative feedback. The easiest way for me to describe negative feedback is to have you think about body temperature in our example. If your body temperature gets too hot, gets too high, we want to negate the rise and bring the body temperature down. If our body temperature falls below 37 Celsius, what do we want to do? We want to bring it up. We want to negate the drop. Negate the drop. So we want to go in the opposite direction of what is actually taking place at that time. And again, all employ receptors, a control center, and effectors to do that. So you're trying to negate the change. If it's going high, bring it down. If it's going down, bring it up. There are some examples, a couple that I want to just mention briefly here, that employ what's referred to as positive feedback loops. What do you think positive feedback might imply? Well, if negative feedback, as we said a moment ago, can be thought of as negating the rise or negating the fall, positive feedback would be to promote the change. Now, it's kind of ironic that we use this term positive feedback as it relates to homeostasis, because really what positive feedback will do for a very short period of time is take us further away from homeostasis. And that might not sound initially like a good thing to do. And it probably isn't a good thing to do for too long. But there are several physiologic examples that I want to mention next that do employ positive feedback. And some examples I can give you here would be things like blood clotting, which we'll talk more about, I think, in chapter 14 coming up in later in the semester. Some of you have had children through vaginal birth, we'll assume. You know, for example, that as the moment of birth arrives, the contractions of the uterus get stronger and stronger and stronger, right? That's positive feedback. Not only does the contraction itself get stronger, but the duration of the contractions get longer, don't they? Yeah. And so these examples of blood clotting and labor pains are both utilizing positive feedback mechanisms to promote for a rather short period of time, hopefully for the childbirth, right? It's not too long. But when you get a cut, for example, and you start to bleed, we know that if it's not a very serious, wound, within a few minutes, the blood has stopped, pouring out of the cut, and we have the platelets grabbing onto the broken blood vessels and perhaps a clot forming, right? The bleeding stops, in other words. The initial process of blood clotting actually does involve a lot of different clotting factors, and these are designed to temporarily kind of take us away from that stable state, but in so doing actually ends up helping us clot. Once the clot is formed, then this positive feedback system begins to kind of tail off. Same for uterine contractions, right? Once the baby's born, after the afterbirth of the placenta occurs, then the contractions begin to decline. It may take some time for that to happen, but eventually they do. Okay. What I'd like to do is sort of wrap up chapter 1 by indicating to you that there are a couple sections here in chapter 1 that I'm not going to hold you responsible for in lecture. So the lecture quiz coming up and the first lecture exam coming up, you don't have to worry about this section 1.6 found on page 20 of your textbook, where it talks about body cavities, body membranes. We're gonna talk more about that in lab, actually this week, coming up soon. However, there is a section here in section 1.6 that I do want you to be responsible for. And this is that section, as I've implied here on the PowerPoint slide, that talk about the various organ systems. And there's a table that appears both in the book and of course here on the slide that lists the 11 organ systems. You should be familiar with those 11 organ systems. You should be familiar with the general major organs that you would find in those systems, as well as their functions. So their anatomy and their physiology. And I think this table does a very nice job kind of giving you a general overview. And so this is something definitely worth spending some time looking at. And I suspect that a lot of you could come up with most of those organ systems and hopefully list the major structures found there and also what the major function is. So again, spend some time in section 1.6 looking over the section entitled Organ Systems. Section 1.7 I did not address here on the slide, but as I'm looking at the chapter here on page 29, this is entitled Lifespan Changes. Each chapter has a section entitled Lifespan Changes. I typically do not lecture over that small section of the chapter, but I would like you to get into the habit of reading it, because what it does is it takes that particular chapter and discusses what occurs in you and I as we get older. It's very interesting. And so I really would like you to spend some time, just takes a few minutes to read over the lifespan change sections of each of the chapters. Section 1.8, anatomical terminology, again, is something you can skip over for lecture. We will be covering it in, I think, exercise 2 of the lab book. So when you come to lab each week, I would really, really like you to get into the habit of bringing your textbook with you. Because, for example, coming up soon when we do exercise 2 in lab, we're going to be learning about body cavities and membranes and quadrants. And we're going to be learning about different surface anatomical structures that are described here in section 1.8. So make sure you please bring your book with you to lab. And I'll also mention as I look through this chapter that at the end of each chapter, there's a very nice chapter summary. They're in page 36 and 37 and 38 for chapter 1. If you don't have time, for example, to read the entire chapter, let's say you have an exam coming up in class, lecture 3 exams coming up next Thursday, and you don't have time to read over the 5 chapters or 4 chapters that chapter 3 or that exam 3 is going to cover. You could go to the chapter summary in the back of each chapter for a quick sort of synopsis or overview of the material. So I would encourage you to spend a few moments, look at these chapters, get kind of familiar with what's there. There's excellent diagrams. The chapter summaries are also quite good. And I think that pretty much wraps up what I want to talk about in chapter 1. Okay? So I'll stop sharing the screen. You should begin to get into chapter 1, start reading it, comparing our PowerPoint slides with what the chapter is presenting. And then if you have questions, please ask those during the review session, which some of you are registered for. I have office hours, 8 office hours a week, and I will have this Zoom room open. I'll also be in my office during those 8 hours. So if you happen to be physically on the 2nd floor of the Allied Health and Science Center, you can certainly come by, or just pop into the Zoom room, the meeting room, and feel free to ask questions. Okay. Okay, we will begin chapter 3 for the next Zoom recording. Chapter 2, remember, as I told you earlier, entitled Chemical Basis of Life. I am not going to do a formal Zoom presentation. There is a PowerPoint that you can download and look at. And don't forget, I think I told you guys about the chapter objectives in Blackboard that you can use as a guide. All the chapters are there, but chapter 2 would be something that you should start to look at and you should begin to study those objectives for chapter 2 and start reading through chapter 2 because we've got that chemistry exam in a couple weeks. Okay, I'll end there, and the next presentation will be on chapter 3.